Okay, so happy Monday. We are going to today pick up basically right where we left off. Last day we were talking about microscopy, and we'll be continuing there and probably talk about a few other things related to studying cells. And we'll also this will also overlap with tomorrow's lab where we'll be talking about the microscope as well. So I kind of wanted to just recap what we discussed so far. So far, we're discussing light microscopes, and we were discussing some things that we can do to improve our images. And there were really three variables: magnification, resolution, and contrast, and different things you can do for each of these different factors to help improve image quality. Magnification, that really has to do with the type of lens you have, kind of how thick it is and the shape of it. Bigger lenses equal better magnification. Resolution, this is really a measure of clarity, and there's a number of tricks for improving resolution. One trick is having a good lens material, so quartz is a lot better than plastic, and glass is somewhere in the middle. We can also use something called oil immersion to improve our resolution, which we're going to talk about oil immersion in the lab in lab 4. So I'm not really going to get into it too much in this lecture. And the last thing is contrast. And what is contrast? Contrast is the ability to be able to see the edge of something. So we can see edges really well by staining them. So I showed you a bunch of pictures that are improving these types of qualities in the images. So today I want to shift over and talk about a special type of light microscopy called fluorescence microscopy. So you can think of fluorescence as a type of staining, but in this case, the stain actually glows. So we can get some really, really nice, very sharp images from these. If you see on the left here, what we're looking at is a fluorescence microscope, and you can see right here, you have a laser. Sometimes they have lasers, sometimes they have UV lights, and the light doesn't go through the specimen. It actually hits the specimen at a 90-degree angle because it doesn't need to go through the specimen because the specimen itself is going to glow. So a normal light microscope, for a good one, you're looking at $5,000 to $10,000. For a cheap fluorescent one, you're looking at maybe $20,000. You're starting to get to more expensive optics, and when you're starting to add lasers and things like that, it gets a little bit more sophisticated. I'm going to talk about this other technique here called confocal in a moment, but let me show you some pictures of some fluorescence microscopy. So there's some pictures. You can see we have different fluorescent stains are being used to highlight different cellular features. So on the left, those little red things, those are the mitochondria. You can see the nucleus here. It's in this big blue ghosty thing, and the cytoskeleton and all these little green things. So some really incredible pictures. I'll show you some more. There's another one. Here's a worm. I'm not sure why they inject it with a green fluorescent dye, but you can see the worm's skin very clearly. Here's another one. Again, I don't really know what they're trying to do other than make a rainbow cell. I thought it was kind of a cool picture, and so I thought I'd show it to you. So let's just talk about this confocal thing for a moment. So one issue with fluorescence microscopy is just like when you're trying to focus on anything. If I'm looking across the room here, and if I'm focusing on the wall across the room, and I hold my hand up in front of my face, my hand is out of focus. As soon as I focus on my hand, the wall is out of focus. And you have the same issue with microscopies. You can't focus everywhere at once when you have a specimen with depth. But you can do that with confocal microscopy. And the way confocal microscopy works is you have those lasers, and the lasers will allow the specimen to glow or emit light at very precise depths. So this confocal microscopy is just kind of a sophisticated type of fluorescence that kind of enhances the image, and then you can build the whole image together in the computer and make a very sharp image. You're looking at $50,000 to $100,000 for a cheap confocal microscope. So I'll show you some more pictures. If you take a look here, this is who is this? National Geographic? No, this is Nikon. and they have a photography competition every year. And if you take a look, it says technique is confocal. That means they're using lasers to get these very, very sharp images. Really, really incredible image of this cell. You can see the nuclei, you can see the cytoskeleton, and all sorts of other features on there. It's a skin cell, looks like. Here's another one. This is also Nikon, and this is another confocal one. This is looking at the surface feature of a tapeworm. So what you're looking at is the head, and those are kind of sucker discs, I guess they're called. Allows them to attach to somebody's intestine. I thought that was a very, very nice picture. Here's another one. This is looking at some brain or nerve ganglia of looks like a chick of some sort. And in this case here, I'm not exactly sure what the technique is, but you can see each cell is coming out a little bit of a different color. So they make out some amazing, amazing details. Very, very cool. As I mentioned, a lot of fluorescence microscopy benefits from digital enhancement. Nowadays with computers and software, you can take 100 pictures and you can use the computer to average them and enhance them, and you can get much sharper details. You can see that right there. Here's a picture using some confocal microscopy, and you can actually build up a three-dimensional image of a lot of things. So this is some three-dimensional mitochondria in a yeast cell, and that's a huge advantage of the confocal stuff. Of course, we're starting to talk about big money, maybe $100,000, $200,000 for some of these microscopes and the very expensive software that comes with them, but you're getting some really amazing details in these cells. Fluorescence microscopy is also used for diagnostics. You can see this is a case where somebody's doing a blood smear looking for malaria in an individual, and the malarial parasites come out in this nice glowing yellow image there. There's another picture. Just wanted to show you there's some really, really incredible pictures of some fluorescence microscopy. So I wanted to just take a quick break here, and I'm going to take some notes. So I started to make some notes here. I'm making a table. You can see I have in the table types of microscopy, and I'm going to talk about electron microscopy in a few minutes. Right now we've talked about light microscopy. So this is the kind of thing I could ask you in the midterm. Compare light microscopy with electron microscopy. Might be a good type of exam type of question. And so first thing to note is make sure you take a look at the words. We have light microscopy and we have electron microscopy. So right at the very start, I'm expecting you to explain what is going on. What is the light part and what is the electron part? And that is actually this first part here I call illumination source. So what's the illumination source in light microscopy? It's visible light, sometimes UV light. So this could be the sun, this could be a candle, this could be a light bulb, this could be a laser, could be all sorts of things that are providing light, usually in the visible part of the spectrum. I will come back to the electron microscope later, but I am going to add one detail here, is that electron microscopes use electron beams. Make sure I spell that correctly. Okay, so light microscopy, what types of lenses did we talk about? So we mentioned plastic on a cheap microscope, glass maybe on a mid-range microscope, and quartz on a more expensive type of microscope. There are a few other, I think, synthetic materials, but quartz tends to be the best material for that. All right, number 3, it says viewing. So what do I mean by viewing? It means usually your eyeball. Sometimes you're using a camera. And you'll see, again, when I get to the electron microscope, what I mean in that there's differences between these things. Light microscopy is in color. Yes, it is in color. We'll say yes, in color. There we go. What are some of the techniques that we've been talking about? So we were talking about oil immersion. Let me just add some bullets there. Oil immersion. We were talking about staining, talking about fluorescence and confocal. So I gotta learn how to spell apparently. And there are other techniques in light microscopy. These are kind of just the ones that we're discussing in today's class anyway. Okay, cost and ease of use. Okay, so the cost is, we'll call these affordable. I know what you're thinking, a $20,000 fluorescence microscope is not affordable, maybe not for you and maybe not for me. I don't have $20,000 burning a hole in my wallet at the moment, but if you think about a hospital, one hospital owning one or two fluorescence microscopes is not unreasonable considering how easy it is to use and how quickly you can get results from some sort of diagnostic technique. Ease of use, affordable, and we'll call these easy to use, meaning that if I have a microscope, I could train you to use it in probably 5 to 10 minutes. That is easy to use. Okay, magnification, again, depends on the exact microscope you have. But usually we're looking at about 100 to 1,000 times for a light microscope. And maybe up to 2,000 to 3,000 times for some advanced confocal microscopes. So I'll say that up to 2,000 times for expensive confocal, we'll call it confocal digital enhancement. Okay, so I have one more value there called resolution. And I'll give you a number. So up to about 0.2 micrometers. So I'm just going to make sure I get the right symbol for micrometer. There it is, micrometer. So we're going to talk about this in the lab in a minute here, or tomorrow, I mean, or sorry, not tomorrow, Thursday. Getting the day of the week mixed up. But let me just talk about a micrometer for a minute. Okay, so 1 millimeter. So I'm just writing on the right side of the screen because I have space. I will erase it later. So 1 millimeter equals 1,000 micrometers. So that's what a micrometer is. And we use this special symbol. This special symbol here is a Greek letter. This is a mu to represent micro in the metric system. So that's 10 to the minus 6 meters. So that's 1/1,000,000th of a meter, 1,000th of a millimeter. So it's a very, very small measurement. And why is a micrometer relevant? Because, well, cells, we measure them in micrometers. And all of your cell measurements we're going to be doing in micrometers this semester. E. coli is about 1 micrometer in size. Animal cell, anywhere from maybe 20 to 50 micrometers. So it's a useful unit of measurement for microscopy. Okay, I'm going to move back to the PowerPoint now. If you didn't get all this, don't worry. We will come back to this table that we're making when we talk about the electron microscopy. So I'll go back to the slides and show you a few more pictures. There's another picture of some sort of brain or something like that. It says here in the live zebrafish embryo using confocal microscopy. So some really amazing pictures. Really, it's just some incredible things done in the last 10 or 20 years with fluorescence microscopes. More and more people seem to be getting them. There's another one up close and personal. So the nucleus is up close and personal, and you can see all of the little yellow mitochondria. So again, very, very stunning picture. Very beautiful, nice colors. Okay, so back to this slide here about what we can do to improve our image. And there's some issues. We have some limits with magnification and resolution. So you may have noticed that in that table I was making, a light microscope, the maximum magnification is about 1,000 times, maybe 2,000 times if you have something really expensive. And you're not going to be able to see a lot of things very well with that. If you're looking at something like E. coli, we can see E. coli, but we can't really see a lot of detail. Same thing with a lot of organelles. Many of them we can't see, or we can barely see any detail with them under a light microscope. So what are we going to do to improve this? We are going to use an electron microscope. So this was conceived of in the 1930s, and actually I believe there were people at University of Toronto that were involved in developing the electron microscope. And the whole concept is that you take a look at a light microscope. Here is the visible light, and the wavelengths are in the 400 to 700 nanometer range. So that means we can see something kind of one-to-one, so in the 400 to 700 nanometer size, right? So I had given you a number which was 0.2 millimeters, which is 200 nanometers. So it's kind of in that range, right? So what if we have a smaller wavelength? So the idea here is, well, let's try a different type of wave. We'll use electron waves. So electron waves are in the picometer range, and which means we can get down and see very, very tiny objects. So here's a light microscope on the left, and we're comparing it to an electron microscope. So notice we have an illumination source. So here's our illumination source. We have a light bulb right here. The light is going to go through, and it's going through the specimen here. And then we have a lens, and eventually ends up with your eyeball, and you see an image. So in the case of an electron microscope, rather than a light bulb, we have an electron beam. And rather than lenses made of glass or quartz, we actually have magnetic fields. So these magnetic fields are used to focus the electron beams. The electrons will either go through or bounce off the specimen. And you're not going to stick your eyeball in there, by the way, unless you want to hit your eyeball with a high energy electron beam. That's not a very good idea. So usually it's captured on film or some sort of digitized computer screen, something like that as well. So I'll show you a little bit about electron microscopy. So the first type of electron microscopy I want to talk to you about is called SEM. That's scanning electron microscopy. So this is actually a photograph of the one that I used at the University of Guelph. And so I want to point out a couple of things. This was purchased in the 1980s. So it was a bit of an old machine, but it worked just fine. I mean, you're looking at $500,000 for one of these things. So want to, if it still works, why sell it or whatever, right? So what you're looking at right here, this here is actually an old computer. What's not shown here is there's actually a desktop computer sitting on the right of this that's hooked up to this microscope. I still use these knobs here, right here. There's some knobs for focusing and whatnot, and I think there's an on button there or something like that. But most of that wasn't actually being used. So this here is the electron microscope. So this thing here, this is the electron gun, we'll call it. There we go. Down here is the sample chamber. So that's actually a vacuum chamber. And so I'll show you how the technique works. So actually, here's some pictures that I took. I took these pictures here. You're taking a look at some Pseudomonas aeruginosa. This is a bacterium kind of like E. coli. You can see up in the top left, you've got some rod-shaped cells. And then these other samples, you can see these ones here. These ones here circled. These ones are dying. You can see they're kind of, the membranes are falling apart. And the blue ones, those are my mutant babies. So those are, that's a genetic mutant that I end up making as part of my research. Here's another picture here. This is something I wish I had done. Lots of people take bugs and they're looking at the eyeballs and the hairs on them, and there's some pretty cool pictures that are being done. So a couple of notes here. You can see on my note it says the specimen is coated with heavy metal. So why are we doing this? Well, heavy elements have lots of electrons, so it's kind of like staining an object. More electrons means you're going to see the object better. So people often use gold or platinum for scanning electron microscopy. Gold is kind of the thing of choice. Why? Because gold is cheaper than platinum, and it's a heavy metal, and a little bit more abundant than platinum anyway. And you can see this note here. It says that in scanning electron microscopy, you're actually getting kind of a 3D surface image. So the electrons hit the object, and they bounce off, and then the computer reads the signal as either intense or not intense, and so you get these different shades. and you get a 3D image. Another thing you may have noticed, these images are mostly grayscale. So electrons don't have colors, by the way. Electrons just have intensity. So high intensity equals kind of light or bright. Low intensity equals dark. We do add color often to, make the image pop out and look a lot more attractive. So you'll see a lot of these images in your textbook, and all the colors have been added false. These are some pictures right here of actually the SARS-CoV-2 virus that's shedding from infected cells. And so they're not really blue or turquoise. This is just to make the image pop out and so you can see the detail really nicely. Here's another picture. These are some HIV virions. These are budding from an immune cell. Again, the color's been added, and you can see it's just popping out and really making it look very, very nice. Here's another one, another 3D image. These are some breast cancer cells that are dividing. You can actually see the nuclei as those big bumps, almost looks like fried eggs. Some more, just some beautiful pictures. I want to show you so many beautiful pictures. There's some E. coli. There's some blood cells. And there's some artsy cells. So not cells, some art projects, salt and pepper. So that's kind of cool. Okay, so let's talk about the technique. I mentioned we add gold to the specimen. This is how the gold is added, this little thing called a sputter coater. I don't actually know how it works. I just basically you put your specimen in there, you press the on button, you come back a minute later and it's done. But I'll show you basically how this works step by step. So there's our specimen. You can see this here is a little mosquito, and it's put on a little platform. It's put in the sputter coater. There it goes. It comes out. You can see it's kind of just a little bit shiny. It has a very, very thin layer of gold on it, which, like I said, is kind of like a stain. It goes into the microscope. It goes into this vacuum chamber. So why a vacuum chamber? Well, air has electrons in it. So we want to pump all the air out to get rid of any interference and hopefully have a nice, clear, and crisp image. Then basically you turn it on and you focus it, and that takes a little bit of finagling. It takes a little more than 10 minutes to train someone how to use this, but I was trained in about half a day to use this microscope, and then you get some images. So in this case, you can see we're looking at the bug's head. So there's the mosquito head. I know what you're thinking. It's so beautiful. Well, probably not. It's an image only a mother could love, I think, from a mosquito. Zooming in, you can see we can zoom into those hairs, and we can just keep zooming in. We can get some amazing magnification from scanning electron microscopy. We'll look at those hair fibers. We can actually make out some detail, and we can even zoom in and see the individual proteins right in the hair. So very, very incredible image from electron microscopy. Here's another image. Sorry if I scared you. This is a hookworm. And, now we know where we get some ideas for science fiction movies, because these things are horrifying. Hookworms just, yeah, that thing scares me. Here's some Giardia. These are little parasites that give you traveler's diarrhea. I think I mentioned these before. They have 2 nuclei, and you can see a bunch of flagella for swimming around. And you can see that 3D image. So that's surface image from scanning electron microscopy. And there's some chromosomes. That's a human karyotype you're looking at there. So 46 chromosomes. You can see, make them out, nice 3D images of these things. So one huge advantage, we can see these small things, and one small thing that we can see that we cannot see with the light microscope are viruses. Viruses are very, very small things. They're in the nanometer range. So if you want to see them, you need an electron microscope. And there's a picture on the left of some Ebola viruses. They're kind of really skinny and spaghetti-like, coming off of an infected cell. And on the right, there is a virus that infects E. coli. It's called a bacteriophage. So there's a second type of electron microscopy I want to talk about. It's called TEM, or transmission electron microscopy. So this one here is very similar to the scanning, except for in this case, you're making a very thin specimen and the electrons are actually traveling right through the specimen. This is very hard on the specimen. It will damage it after a certain amount of time, but you can get a different kind of image from a transmission electron microscope. In this case here, you're getting an image of inside the cell. So I did have the fortune to be able to do a little bit of transmission electron microscopy, sort of. It turned out that the training would have taken too long, and most of it was kind of done for me. These are my samples. They didn't really turn out that great. You can see inside of E. coli, all these bumps are probably ribosomes, and you can see the membranes are falling apart. My samples weren't very good, and in the end I didn't do it again. It just took too long, and I went on, moved on, and did something else. But there's my image, so I thought I'd share that with you. Here's a professional image of E. coli, and this one has also been colorized. Oh, sorry, this isn't E. coli. This is a different bacterium called Corynebacterium diphtheria. That's the one that causes diphtheria. So in this case, we can see inside cells, which is very useful for looking at organelles. In this case here, you can see they're trying to show you can actually get down and see membranes. You can actually see the bilayer of a membrane. So this is a very, very small thing to see. Here in this case, we're looking at two cells that are side by side, and you can see two bilayers, and you can actually see the little layer in between the cells called the intercellular matrix. So amazing amount of magnification from transmission electron microscopy, even better than scanning electron microscopy. So here's a colorized image. This one's pretty cool. You can see the endoplasmic reticulum. You can see ribosomes all over the place, and the big red things are mitochondria. Some incredible images. Here's kind of a comparison of the transmission and the scanning electron microscopy of both the endoplasmic reticulum. So you're getting different types of detail from each of these types of microscopy. Okay, so there's our light microscopy. There's some bacterial cells. There's the scanning, much closer, much better detail. And there's the transmission. So you're getting to look inside. So I'm going to switch back to this document here. And this was our document comparing our light microscopy and electron microscopy. So I already have the first part filled in for electron microscopy, in that we're using electron beams. And what are we using to focus those electrons? We are using magnetic fields. Viewing, you're not using your eyeball, but you might be using photographic film. So that's the classical way. Up until nowadays, computers are so much more common that we can use digital detectors and directly form a digital image. So I'll say that, photographic film and digital cameras. There we go. Is it in color? No. Electron microscope images are in grayscale. We can put a note, color can be added later. So you color whatever color you want, if you want your cells blue or green or pink. Just depends on what kind of thing you're looking for. Techniques, again, there's many techniques, but I am just going to put the two that we talked about. So SEM, so that's scanning electron microscopy, and TEM, that's transmission electron microscopy. Okay, cost. These things are expensive. You might be able to get a used one for $100,000 kind of thing, but you're looking at $500,000 for startup costs usually, to possibly many more dollars to get to use. Scanning electron microscope, half a day training, transmission more like a week. So requires training, we'll say. Requires training. Okay, magnification. So SEM, I'm just trying to remember the values here. SEM is up to about 200,000 times. And TEM is up to about 1,000,000 times. So we can really magnify our specimens to an incredible amount. And then resolution is up to about 2 nanometers. So what's a nanometer? A nanometer is 0.001 micrometer, in this case 0.002 micrometers. So let's just get our symbol in there. There we go. So that's about 0.002 micrometers. So basically you can see membranes. You can't really make any details on the phospholipids, but you can get right down there and see the details on a lot of organelles. So I thought I'd put this together for you. I know there's a table in the notes that may have some ask for some different information, but hopefully this is enough to get you started to fill that in for study purposes. Another really important thing for these types of questions, if this was a midterm question, is that I expect you to give me examples. So don't just say, well, under a light microscope you can see a cell. Oh, and under a scanning electron microscope you can see a cell. Okay, that's great. That's just not very specific. Give me an idea of what you're looking at. So a light microscope, you're looking at a living cell in some cases, or let's say a gram stain, or something you would actually look at. Maybe you're looking at chloroplasts. That's something we'll see in this week's lab. We'll see some chloroplasts, for example. A scanning electron microscope, right? There's some particular features around it. You're looking at the surface of a cell or the surface of an insect eyeball. In transmission, you're looking at organelles, so the internal structure of a cell or something like that. So make sure when you give examples that you are specific. It's very important in science classes that you are specific when you're discussing things. Okay, so I'm going to go back to the PowerPoint now. Hopefully everybody got this. If not, you can review the video once I get it posted. Okay, so, oh yeah, I wanted to show you this. This is apparently the world's largest transmission electron microscope. I don't know how much it costs. You can call it Patashi if you want. They're in Japan. But if you take a careful look at those are railings. So this thing is like 3 stories high. It's massive. There's a guy, he's using it. He's probably paid a lot. For transmission electron microscopes, there are not a lot of really skilled technicians out there. And so, you know, these people are paid decently because often they're doing what only 10 people in the world can do in some cases. You can see a whole bunch of computers and hopefully he's getting some nice images. If that doesn't work, I found this one on a few years ago on the internet. I can't find this website anymore, but it was $50,000 apparently. So if you're looking to start up on the cheap, you can maybe find something used. So light microscopy, electron microscopy. Make sure you're using keywords, light, right? Light microscopy, electron microscopy uses electrons. So don't forget to use those words if that ends up on the midterm somehow. Okay, so I want to talk about one other type of microscopy that's kind of newer. This was invented, I think, in the 1980s. Although up until about 10-ish years ago, you actually, if you wanted one of these, you'd have to build it from scratch. Although now you can actually buy them. These are called scanning probe microscopes. There's a whole bunch of different techniques and they have different acronyms. You'll see some of them on the slides. And these don't use a traditional illumination source. These use a probe. So you can kind of think of this as like a blind person. So a blind person may walk along and you know, they're using their fingers to feel what they're touching. So maybe they come up to a tree and they feel the bark and the branches and the leaves. In this case, what is happening is the probe is scanning. So what the probe is, you can think of it as like a finger, and it's just going back and forth very carefully and systematically and feeling every bump. And every bump is registered in the computer and turns into a 3D type of image. So I'll show you some images. This is really easy to do on hard surfaces, so like metallic surfaces. And here's an example using something called STM. That's a type of scanning probe microscopy called scanning tunneling microscopy. And this is a gold surface. And you're probably wondering what you're looking at there. Those are actual gold atoms. So this type of microscopy can actually get down and you can actually see atoms. So this is really incredible stuff. Here's another picture. This is a silicon surface. Sometimes you get nice pictures, sometimes they end up being a little fuzzy, kind of depending again on the software, and a lot of people have had to build their own instruments from scratch, so that's pretty impressive to begin with. Another thing you can do with this type of microscopy is you can actually pick up atoms. You can pick the atom up and you can move it. And this is something IBM did. So can you see it there? IBM made their corporate logo with atoms. I can't remember if this is silicon or gold, but there it is, IBM. So pretty cool stuff. I was going to show you this. So IBM made their corporate logo here. And then a few years later, they decided, well, let's go an extra step and we will make a video. So this is called A Boy and His Atom. I'll play this for you. Oh, how come that's not playing? There we go. And the volume down there, and they're just talking about how they're moving atoms frame by frame to make this little video. I won't play the whole thing for you, but you can watch the beginning part, and if you want to watch the whole thing, just Google it, you'll find it, it's on YouTube. So here goes. There's the little atom, and it's like a ball, and there's a little boy they made. So pretty cool. And there he is dancing, and if you watch the whole video, you'll see he plays with his ball and has lots of fun. So I thought that was a pretty cool video. So here's one that you can buy, and this is a commercially available one now. Like I said, maybe 10-ish years they've been out. This one is claiming to be the world's fastest, so I guess that's a good thing. And there's another acronym for you, atomic force microscopy, AFM. And you can see all these atoms in this surface. So it says there's atoms of tin, lead, and silicon on that surface. So we can actually take a look at molecules, which is really incredible. Here's one more picture I thought I would show you. It's a little bit more difficult when you get to biological specimens. So it turns out biological specimens, as you probably know, they're not hard usually, they're kind of squishy. And so if you're, just again, think of the blind man, if you're touching something squishy, just by touching it, it's gonna get mooshed around. So like Play-Doh or things will get bent. You're touching a flower, the leaves and the petals will bend over. So sometimes it's a little harder biologically to get good images. So we can't quite get down to atoms when we're looking at biological specimens. But we can get some interesting details. Here's some pictures here. On the left, we have some red blood cells. On the right, we have a platelet. So you can see getting a little bit of detail out of this. And probably as people get better at this in a few coming years, we'll get some more interesting images. There's actually a herpes virus on the left, and you can actually make out the individual proteins on the surface of it. There's some E. coli on the right, so some interesting images, and we're probably, like I said, going to see a lot more of this in the research in the next coming couple of decades. There's some more pictures, human chromosomes, breast cancer tissue, and there's E. coli again. And this is one of my favorites. You can see on the left is the computer-generated image of the DNA double helix. And on the right, you can see the atomic force microscope image. So you can see as it goes back and forth, there's a little bit of distortion in the image, but you can actually make out the helix of the DNA, which I thought was really, really incredible image. Okay, so where are we with time? We are at 4:40. I have a Kahoot for you, just 3 questions. And so load up your app and give you a chance to get a little bit of feedback. So here we go. Okay, give you about another 30 seconds to join. Okay, so here we go. Topic 3, microscopy. What is resolution? Okay, so most people got the green one, which is how sharp the edges of an image appear. And that is actually not the right answer. That is more to do with contrast. Resolution is a measure of clarity, or another way to think about clarity is the ability to distinguish that two objects are distinct objects. So one person got it right, and so one person, Shane, got 700 points. Well done, Shane. All right, question 2. Which of the following matchups is false? All right, the green one is correct. So good job. TEM stands for transmission electron microscopy. So good job on that one. Shane's probably still in the lead. Got the extra bonus points from the first question. All right, last one. True or false? These types of microscopy are correctly ranked from lowest to highest magnification. Okay, the correct answer is false. Let's just take a look at that image. We have probe microscopy, TEM, SEM, fluorescent, and compound microscope. So we're going here from smallest to largest. So it's false because it's asking about the other way around, lowest to highest magnification. So the highest magnification there is the probe microscopy. So good job if you got the false. And we'll see how the scores come out. Kyla, Boss, and probably Shane. No, wow, out of nowhere. Good job, Rianne. Okay, so I'm gonna go back to the PowerPoint. I realize that we're running out of time here. But I want to discuss a couple other things and then I'll let you go. And the rest of the topic I will just leave till last day. So as I mentioned, this has been a pretty typical kind of midterm question. I have a few of these around microscopy. You can see this particular one is actually asking you to compare light and electron microscopy. And it does distinguish and say, you know, describe both SEM and TEM. I could easily make a question comparing light microscopy, electron microscopy, and probe microscopy. This one's open-ended. It's not telling you specifically what aspects to compare, but I would suggest that the ones that I made on that table are important, particularly talking about light and electrons and what the lenses are. Very important to give examples. And this is worth 5 marks. So each point would be 1 mark. So let's say you have a table and you have illumination source, and so light and electrons would be that would all be 1 mark for kind of, you know, that 1 point. If you're vague, if you talk about how things under an electron microscope look bigger, I don't know if I really give you a mark for that, but give me information, just saying you can see objects down to the nanometer range. You know, now you're starting to get into the details. So we'll talk more about midterm type questions as we get closer to the midterm. We're getting there. This is topic 3, and the midterm will be after the plans or after topic 6. So we're kind of halfway there towards the midterm. So I'm going to actually stop there for today because other than that, I mean, I have a bunch of other stuff to talk about and there's no way I'll finish it in 5 minutes. So I guess we're done a little bit early today. Hope you have a great Monday. If there's any questions, let me know. If not, I'll end the slideshow momentarily here. Okay, well, I suppose that's it for today. So hopefully you have a wonderful afternoon, and we will see you on Wednesday.